Good morning, everybody. Thank you very much for all of you for joining us today on this webinar of Cultivating Precision Medicine, Leveraging Biobanking Omics Projects to Accelerate Biopharmaceutical Discovery and Development. Very excited today that we're going to be able to have presentations from four biopharma experts in this field, and to be able to kind of present pharma's perspective on the use of biobanks and the use of omics data and how it's integrated into their discovery projects. Just to give you a quick rundown, if you don't know who CQDM is, CQDM is a biopharma consortium in Canada. We really look to fund early stage collaborative research in partnership with our global pharma partners, and then and with other industry partners. The idea really is to help de-risk early stage research and move it forward so that we can, develop cures and solutions to unmet needs in the health system. We have 9 active pharma members, including some of the groups that are talking today. So Sanofi, Merck, and Amgen. Decode Genetics is an Amgen company. One of the roles that CQDM plays is to be able to facilitate interactions between our global pharma partners and the Canadian ecosystem. And so events like this today, where we are bringing a pharma perspective on omics and biobanking, is an integral part of that. In addition, tied to this webinar, in the afternoon, we are doing a kind of pitch day for biobanks, for Canadian biobanks. And so it'll be the other way around, where the Canadian biobanks that have been selected by our pharma members will be presenting to our pharma partners. And so we're hoping to facilitate interactions and discussions and really be able to bring Canadian research and the excellence that we do here to the global stage. So thank you for the biobanks, if you're on the line now, that have applied. We really do appreciate it. And thank you to the groups. We're looking forward to hearing from you this afternoon. So we're going to start today's webinar with a pre-recorded presentation by Kari Stephenson of Decode, but then he'll be able to join us quick after for a quick Q&A. So if you do have any questions, feel free to put them in the chat room, and we'll be reviewing those, and we'll be selecting some to ask to Kari. So if we can have the video now from Kari, please.
Thank you for inviting me here today to talk to you about precision medicine. And keep in mind that the key to the understanding of human disease lies in the study of the biologic foundation of human diversity. And I'm talking about human diversity as broadly defined, including education, socioeconomic status, and things of that sort. And precision medicine is simply medicine practiced in the context of understanding of the molecular nature of human diversity. And human diversity begins with diversity in the sequences of A, C, Gs, and Ts in the human genome. And just on the basis of looking for the diversity in the sequence and looking for correlations with clinical phenotypes, we at Decode Genetics in Iceland have discovered a very large number of common variants in the sequence of the genome that correlate or associate with a very, very large number of diseases. And keep in mind that the common variants in the genome are the variants that confer on us the human diversity broadly defined. And even though each one of them confers a very small risk of a disease, you can have a confluence of these common variants that confer very large genetic risk, even though they account for very little heritability. And these are the variants that came with us out of Africa or have been under positive selection, or both. We have also found a large number of rare variants in the genome that confer large risk of disease. And I'm absolutely convinced that there are a lot of rare variants in the genome that confer a little risk of a disease, but we cannot simply find them because we don't have the power to do so. But these rare variants are either new, or they are under negative selection, or both. But there is more to human diversity than just diversity in the sequence of the genome. The environment does have a significant impact on human diversity, mostly by interacting with variants in the sequence. And we have grown accustomed to look at evolution as consisting of interplay between random mutations in the genome and then selection by the environment of attributes that are rooted in the diversity in the sequence. But there is more to the environment than that. We are under the influence of the environment from the moment we are conceived and until we die. And actually, the older you get, the more opportunities has the environment had to have an impact on you. And you can basically look at time or age as a funnel that collects environmental influences. And most of the diseases of man that have significant public health impact are diseases of relatively late onset, diseases that happen because the environment has had significant time to opportunity to have an impact on you. So we have the task at hand, which is to bridge this gap between the influence of the genome through the diversity in the sequence and the clinical phenotype. And this gap is filled by all kinds of environmental components: diet, education, exercise, socioeconomic status, and so on. And it is relatively easy, in a way, to pick out the genetic component to the risk of diseases. You just sequence very large number of genome of people of all kinds of ethnic backgrounds. It is more difficult to figure out how to systematically study the environment. But we and others have started to do the following. In addition to looking at the diversity in the sequence, we have been looking at the amount of messages, amount of RNA, splice form of RNA, level of protein in blood, isoform of the proteins, and then bring all of this together to the disease or the clinical phenotype. And when it comes to looking at, on one hand, at the proteins, on the other hand, at RNA, the protein have a substantial advantage over, or basically the proteomics a substantial advantage over the transcriptomic, because to look at the transcriptome, you basically have to have a biopsy from the organ of interest. But we are so lucky that a very large percentage of the proteins in our body make it to blood, and in blood they are in some sort of dynamic equilibrium with the protein in the tissue where it does its work, and therefore it allows us to look for correlation between the levels of the proteins in blood and diseases. And keep also in mind that the proteins are the business molecules in our body that make everything else. And now I've told you that you can look at time or age as a funnel that captures environmental influences, and I've told you that we can use the proteome to capture some of the environmental influences. And if both of these are true, you would assume that there is a correlation between age and level of blood, proteins in blood. And indeed, what I'm showing you here is when we have developed an algorithm to predict age on the basis of level of about 1900 proteins. And when you do that, and you plot the predicted age, or the biological age predicted by proteins, against the chronological age, the correlation is very, very good. The R-squared is about 0.96. And it's very interesting that you can predict the age on the basis of proteins, because when you begin to see a departure between chronological age and biological age, it means that something bad is happening. If your biological age exceeds your chronological age, something bad is happening. And we can also use the proteins to predict the age of individual organs, which is provides a fascinating opportunity for measuring health of specific organs. So basically, by using the protein, we can begin to bridge the gap between the genome and the clinical phenotype. And what is particularly interesting is that the proteome gives you an opportunity to capture time. When you're looking at genetic risk, you're basically using the variants in the genome that existed from your conception to your death. But when you look at the proteins, the level of proteins in blood, they rise and they fall as a function of time to and from events. So they allow you to capture time, they allow you to use the proteins as biomarker, not just of progression of disease, but also of regression of disease. And let me now give you one example of a level of a protein in blood that correlates with a disease, where the variants in the genome that affect the level of the protein also correlate with the disease. And the example I want to give you is in the cordon-like 2 locus. There's a variant in the cordon type 2 locus that correlates with risk of colorectal cancer. It confers an odds ratio of 1.32. But this variant does not correlate with level of transcripts. So when we looked at the proteomic data, we could demonstrate that this variant correlates is the variant that has the biggest impact on the level of the cordon-like 2 protein in blood. And what is interesting is the cordon-like 2 binds to BMP2 and prevents it from binding to BMP2 receptors. And it's interesting because it has been known for a long time that loss of function mutation in one of the BMP2 receptors predisposes to juvenile polyposis coli, which is a precancerous condition for colorectal cancer. So we assume that the cordon-like 2 is an important contributor to the pathogenesis of the colorectal cancer in these cases. Then I'm going to give you one example of a level of protein in blood where the variants in the genome that correlate with the level of the protein do not correlate with the disease. And the example I'm going to give you lies in the beta defensin 4A, which has a dramatic increase in level in blood in patients diagnosed with psoriasis. The increase is about 0.86 standard deviations, and actually if you take all of the 62 variants in the genome that have been shown to correlate with risk of psoriasis, they correlate with the level of beta defensin 4A. However, if you take the 7 variants in the genome that have been shown to affect the level of beta defensin 4A, they do not as a group correlate with risk of psoriasis. So our assumption is that the level of beta defensin 4A is altered as a consequence of the disease rather than participating in the pathogenesis. Another thing we can do is to take a large number of proteins in blood and put together algorithms that allows us to use the level of this protein to predict phenomena, biological phenomena. And basically, when we take about 100 proteins in blood, we can use them to predict how much is left of life for people who are middle-aged or older. So what we had basically is a plasma sample from about 23,000 individuals that were recruited about 20 years ago, or between 15 and 20 years ago. And in that group, 7,400 individuals had died when we had an opportunity to measure these proteins. And we could use these proteins in one of our machine learning algorithms to predict how much these people had left of life. And we could find 5% of people between the ages of 60 and 80 who were about 88% probability of dying within the next 5 years, and another 5% who were in the same age range were at virtually no risk of dying within the next 5 years. So we could basically, on the basis of this, make these predictions, and we happen to have very large amount of phenotypic data on a large number of people on whom we had measured protein, and we could show that this algorithm correlated with all kinds of physiological phenomena. So the shorter life you were predicted to have left, the weaker is your hand strength, the worse you perform on exercise tolerance tests, and the worse you perform on tests of cognitive function. So this turns out to be an assay for human frailty in general, and what is more, we have shown that simply by treating certain diseases, we can diminish the probability of all-cause mortality substantially. So this can serve as a biomarker, not just of human frailty, but also a regression from human frailty upon treatment. A very important aspect of our ability to look at the human diversity as conferred on us by variants in the sequence versus conferred on us by the environment is the possibility of separating these two forces in specific conditions. And what we did is that we took a polygenic risk score for cardiovascular disease, for coronary artery disease, and a proteomic risk score for coronary artery disease in a group of people who are 56 years of age. And what is particularly remarkable is that the polygenic risk score could capture a hazard ratio of 1.21, whereas the proteomic risk score captured the risk of hazard ratio of 2.27. So the proteomic risk score could capture much more risk. And why should that be? Because basically, when you're doing the polygenic risk score, you're working with variants that have been in your genome your whole life. So you're predicting lifetime risk. But when you're looking at the proteomic risk score, you're probably not capturing true risk. You're documenting early steps in the pathogenesis. But what makes this even, and basically when you're 56 years of age, most people have some atherosclerosis already ongoing, so you can capture the consequence of that rather than the prediction of something that may happen in the future. But what makes this even more interesting is that these two risk scores, the polygenic risk score on one hand and the proteomic risk score on the other hand, are uncorrelated. And that is fascinating because that basically means that once the atherosclerosis starts, there are forces that are separate from the genetics of onset that begin to influence the progression. I think that if we would have done this in a group of people with an average age of 35 years rather than 56, the polygenic risk score would have captured more risk than the proteomic risk score, and because the proteomic risk score would simply have been a surrogate for the genetic risk score, and this would have been completely correlated. But this means basically that this gap between the genome and the clinical phenotype is at times unbridgeable, and which is fascinating because then we will have to begin to look at the proteins that correlate with the presence of a disease, where the variants in the genome do not correlate with the disease, not simply as passengers. They could be participating in progression even though they do not participate in onset. But I told you that I was going to discuss precision medicine. And keep in mind that precision medicine is simply medicine practiced in the context of understanding of the molecular nature of human diversity, molecular understanding of the type that I have been discussing up until now. And basically our ability to discover the molecular mechanism of human diversity is substantial. But in our discovery work, we are always trying to figure out what characterizes a group of people who share a phenotype, share a disease. In precision medicine, the task at hand is to figure out into what group does the individual fit. And how can we make it possible for the population in general to take advantage of all this understanding of the molecular nature of human diversity. But once we begin to work on precision medicine from this understanding, from the point of view of this understanding, when we begin to fit an individual into very large number of groups like this, when it comes to risk of heart attack, risk of cancer, risk of all kinds of diseases, there is an extraordinarily detailed information that is collected about the individual, who the individual is, and more importantly, who the individual is likely to become in the future. And it is very likely that people will feel uncomfortable about having data of that sort in the hands of healthcare institutions, or whatever institution. But it is also likely that people will feel much more comfortable having those data in their own database. So I think, and what's interesting when you look at that in the context of the development that has been taking place in the healthcare system over the past couple of decades, is that people have started to take more and more control of their own healthcare. Almost everyone Googles the disease before they go to see a doctor. People are buying all kinds of wearable devices to document their sleep, their movement, etc., buying blood pressure monitors in the drugstore, glucose monitors, and then they become subscribed to services like 23andMe. So I think that the way in which we will probably implement precision medicine in the future is to make available to the customers an analytical platform onto which they can download all kinds of information on their own healthcare, electronic medical records, medical images, family history, health history, medication, diet, data from wearable devices, and even genomic data and proteomic data bought from somewhere. And the analytical platform is probably going to be AI-driven, and it will be able to generate proteomic risk score, polygenic risk score, document biological age of the individual, biological age of individual organs, match signs and symptoms with side effect of drugs, analyze medical images, provide preliminary diagnosis for new signs and symptoms that are put in the context of the history of the individual. And this is going to lead to better care of patients, more effective maintenance of health, but most importantly, it's going to empower the customer, to become a custodian of his or her own healthcare, become custodian of their own medical information, and it's going to generate healthcare without borders. So I, living in Iceland, can have basically the same foundation, or have a foundation of my healthcare that is the same as people have in places like Canada. So basically, the key to the understanding of human disease lies in the study of the biologic foundation of human diversity, and the key to the healthcare of the future lies in taking advantage of our understanding of the very same human diversity. And thank you for your attention.
Thank you very much, Kari. Pleasure to have you with us today.
Thank you.
So, start with a quick question. And to the participants, I apologize. We're working on it. I know there should be a place to submit questions. We're working on getting that activated. But in the meantime, there were some questions that were submitted with the presentations, and so, or with the invitation. Sorry. And so we can start with some of those. So, you touched on it a little bit, but you talked about how, there are single variants, that you think that there are a lot of single variants out there, but we didn't have the power, or we don't have the power to find them. Are we getting to the place where we can start to find the single variants, that the low-hanging fruit that we found previously is now, taken, but there's still lots to find, in the human genome?
I was talking about rare variants with small effects.
Right.
And we probably have power to pick some of them up, and the power is increasing. But what I think is even more interesting, that we have now discovered ways in which we can find interactions, epistasis between variants. That has been something that has been eluding us in the past. First of all, we have now sufficient material to pick up the interaction, and then we have also figured out little tricks that allow us to pick up epistasis, and also interaction of sequence variant with specific components in the environment, and that I think that are particularly exciting. And I can tell you one little anecdote from that. You may remember that, a few decades back, it was the prevailing opinion that modest consumption of alcohol could protect you against cardiovascular disease. Then it was subsequently shown that that is probably not true, right? But now we have discovered that basically that there is a missense variant in alcohol dehydrogenase 1b, which is such that if you have that variant, your non-HDL cholesterol is your level of non-HDL cholesterol is inversely correlated with your consumption of alcohol. So the more alcohol you consume, the lower is your non-HDL cholesterol, and that actually translates into less risk of coronary artery disease. So basically, the old story was correct in the context of a sequence variant that has an allelic frequency in Europeans that is about 2%. So you can show clearly in that case the interaction between a specific sequence variant and the effect of alcohol. Right. So now the only thing you need to do is to have yourself genotyped, and if you're lucky, you can just begin to drink from morning to night.
The powers of science. I know that Decode was founded on a the Icelandic kind of population and a biobank and data there. But I mean, Amgen is a global company, and I think doing a lot of work. Is there are there challenges? Are you working with outside biobanks or outside data access, and what's the challenges of kind of integrating that type of data?
We have in our research, we have biological sample from 2.5 million people, and only 180,000 of those are from Iceland. Right. So we are working with healthcare information and biological samples from all over the place. We, like others, have been somewhat negligent when it comes to gathering, data and biological samples from people of non-European origin, but we are beginning to increase that, and that is one of the things that I expect will contribute to new discoveries in the near future. Right. Because basically, if you look at the locus with a variant that correlates with a disease, you can basically assume that that in the African genome, there's going to be 3 times fewer correlated variants in that locus, which gives you increases dramatically your ability to find map and determine exactly what the gene is. Right. That is affected by a variant. So there are a lot of little things like that that are awaiting us in the near future.
Okay, wonderful. Well, thank you very much, Kari, for joining us today. It's been a pleasure talking with you, and thank you very much for the presentation.
Thank you.
So next we'll be hearing from Simone Longridge from Merck. Simone leads a discovery group at Merck in the data AI and genomic sciences. Her group leverages diverse human genetics and genomic resources for precision medicine drug targeting in discovery and validation. Simone?
Hi, thank you. Can you hear me okay?
Yeah.
Perfect. Well, thanks so much for the invitation to chat here a little bit about, what we do in our group, especially to a Canadian audience. As a Canadian myself, it's a rare opportunity I get to talk, to a mostly Canadian audience, and very much appreciate that. As described, our group is really involved at Merck, which obviously is a large pharma company invested in developing drugs for a range of generally common diseases. Our group is focused mostly on the non-oncological diseases that cross neuroscience, immunology, and cardiometabolic disease. And we are in early discovery, which means that we're tasked with finding potentially novel targets for therapeutic intervention. And of course, as mentioned, we are using human genetics and genomics to do that. So let's click on the slide here, see if it will move. So, of course, to leverage human genetics and genomics, wanted to, with the recent excitement as well, always when something new comes about, was re-invited to go and look back at the Human Genome Project as a whole and sort of the history of its development over the last couple of decades, beginning in 2001 with the first draft of the Human Genome released, with the work of the International Human Genome Sequencing Consortium led by Francis Collins, and of course the Celera led by Craig Venter at the time, and very exciting to have this first draft of the human genome to be able to understand, sort of, as Kari already pointed out, the possibility of understanding human genetic variation and how it is linked to all kinds of human traits, et cetera. And so that, of course, was followed in 2003 by the very first reference genome that we use, and actually to understand how variation itself exists against a reference. Of course, you need a reference to compare things against too. But really that was, the first reference genome that was still missing quite a bit of sequence. So really at that time, they estimated we had about 20,000 genes from the original draft that guessed at about 30 to 40,000. But even then, with that new draft, we still have 150 megabases of unknown sequence that contains a lot of repetitive DNA that we didn't understand. Finally, over the next decades, with the evolution of new sequencing technologies, with being able to read longer regions of DNA, and also scale bioinformatics that allows us to analyze this large amount of data, we were able to, by 2020, reach what we call the first full genome sequence, called the T2T, which means telomere to telomere, where we filled in a large number of these gaps that were unable to be sequenced previously, that included a lot of segmented duplications, parts of chromosomes that also included a lot of repetitive sequence and so forth, and to also find things that really didn't map to the human genome, so things that were more insertions of novel DNA. And then even now, as I was mentioning, the excitement of the early part of this year was that, you know, of course, the original T2T and the draft before that was based on essentially a single person's human genome. But we all know that no single genome can represent the entire genetic diversity of our species. And so it's very exciting to see the draft pan genome now that includes, you know, up to 47 different individuals of various descents from Africa, Americas, Asia, and Europe, and allowed us to identify a lot of regions of the genome that are polymorphic across different populations of people. And as Kari already alluded to, this is important for understanding, of course, the genetic diversity that we then link to, you know, various traits in humans, including disease. So obviously, as a company developing drugs, we are interested in disease-associated genetic variants that help us to identify potential new drug targets. And the way that we do that are by genome-wide association studies, where we group people with and without disease and associate them to genetic variants, you know, just traditionally shown in these Manhattan plots, where we have the chromosomes along the x-axis and the statistical significance of a genetic association with disease on the y-axis. But of course, we all know that, you know, there's a large peak of variants that are associated with a locus in the there, and we don't always know exactly which of these variants is necessarily the causal variant linking it to disease. So this large collection of variants, just on the right now, we have an ever increasing number of associations of different genetic variants with disease, up to more than 55,000 in the GWAS catalog, which is kind of catalog of all disease gene associations or all disease variant associations, across all of the chromosomes in humans. And so one of the next challenges, I would say, is, you know, is how we link, as I mentioned, each individual variant to being causal. And also the challenge still remains to link each of these single genetic variants to a gene. So we can make a best guess, but we like to use statistical methods to make our guess better. And so we use a statistical method called fine mapping in order to take a more educated guess at which genetic variant might be causal for a particular trait. And then, of course, we need some additional data to understand which the most likely gene under a particular GWAS peak is actually the gene linked to that causal variant. And so what we really need is additional data, which already is super nicely and definitely hard to follow Kari's talk on that, to understand, you know, especially for protein coding genes, you know, for which protein coding gene is a variant actually affecting. And so we use both gene expression by transcriptomics as well as protein abundance to link those particular human traits that can be measured in blood or other serum, biological fluids to genetic variants, and then link those as well via a process called co-localization, which is another statistical method that can link both disease associated genetic variants with things that are linked to gene expression or protein abundance under the same set of variants. This is basically the process of integrating GWAS with multiple functional genomics to really connect variants, genes, and proteins to human traits for target discovery, but also, you know, as Kari mentioned, to different elements of disease that includes progression. Okay, so next slide, you know, just one example of what I just said is the example of the FTO locus that was linked with obesity quite a while ago. So amongst individuals of European ancestry, the FTO locus had this greatest effect on obesity at the time and is really still the most common solid causal link, most common link from a genetics point of view to obesity. And you can see that it replicated across multiple ethnic populations. The Europeans, the Asian and African populations also all showed a GWAS peak over the first intron of the FTO gene. And despite the fact that each included different individual variants unique to those populations. And in fact, though, looking at the biological side of the link of this FTO locus with obesity, this link is more difficult to establish. And in fact, number of studies over the course of many years have failed to establish this causal link. In the meantime, we've used other methods of looking at the epigenome. So that is the regulation of our genome governed by histones, chromatin modifications to govern actually expression of genes. And here, epigenetic methylation of the associated region in the FTO promoter was actually shown in 3D space of the nucleus to impact not necessarily FTO expression, but expression of neighboring genes, IRX3 and IRX5. And now would say that these would be the candidates to investigate with respect to obesity. Okay, so you know, all of these examples are really representing advances in our ability to detect variants in the genome, and combined with the GWAS and other human traits are really allowing us to develop these associations between genetic variants and a particular trait in a human. And we have different kinds of variants. We went over sort of, you know, some of these already with Kari's talk, where we can detect very common genetic variations with low effect size, all the way to very rare variants that we can detect by either whole exome or whole genome sequencing that have potentially a higher effect on a genetic trait. So here, just giving one example again, we have the example of the variation in the LPA gene. LPA as a gene product is lipoprotein A. Similar to LDL, lipoprotein A is added to a component that also contains ApoB and a LDL-like particle that contains cholesterols and other lipids, and assembled into this particle. I don't know if you can see my little cursor showing the actual particle here. But essentially, polymorphism in the LPA gene encoding this protein with the large number of repeats here explains 30 to 70% of the variability in the plasma concentration of LPA. And a large part of that variability, it's found out with different now modern genetic associations, is established by the variable number of tandem repeats within the one of these repetitive regions that is part of this gene. And as you can see on the right, a very recent publication was, you know, really finally able to dissect the contribution of the number of repeats now detectable with these new methods and, you know, higher resolution genetic sequencing to, you know, really build a model of how the number of repeats in this particular genetic region predicts plasma level of LPA, which is a very strong risk factor for development of cardiovascular disease. And so here you can see with the number of repeats, you have lower LPA, the fewer repeats, higher levels of LPA. And just another example of how genetics, especially in this case, can allow us to predict the level of protein that is a severe risk factor for cardiovascular disease development. And here, especially important because this gene is not conserved in any other animal species besides old world monkeys and hedgehogs for some reason. Okay, so so here, you know, obviously we're in the business of developing drugs using human genetics, and we do that in our group because we actually have some evidence that it's helpful to use genetic evidence for drug development. We have probably always seen these papers where we have from 2015 onwards from Matt Nelson et al. Showing that if potential drug targets are supported by human genetics evidence, they have 2 to 4-fold increase in the probability of success for going through to the final stages of drug development. This is dependent sometimes on the effect of the variant. In some cases here with the more rare variants that are represented by, for example, OMIM and Mendelian disorders, have an odds ratio between 5 and 10. for increased success, whereas those coming from more common variants have a somewhat lower, but still overall positive success rate for impacting drug development. And this continues to be true even for 2021 FDA approved drugs. 2/3 of them are supported by human genetics evidence. That said, there are still a lot of challenges, some of which I've outlined below already. And we are really, from this nice review from Tam et al., really at the tip of the iceberg of what we can do, as also gone over by Kari, and what we can investigate in being associated with genetics. So here we have, a lot of, as I said, GWAS data supporting gene or variants to disease associations. But we are still missing a lot of the interpretation of those variants in terms of their function biologically. And so we need all of these elements under the tip of the iceberg in order to help us do that. One is to, as we've already pointed out, to have larger, more ethnically diverse reference panels, which we have achieved earlier this year. But we still need to also collect a larger number of individuals with disease and genetics in order to understand the genetic variation there that's associated with disease. And so here, essentially, we need more data to support this. And of course, some of these are on this dark side where I think a lot of that can come from biobank data of that includes deeper disease phenotyping and other biological measurements we can do, such as plasma proteomics and transcriptomics. So one example of a biobank that we leverage is the FinGen consortium that we're part of. This is a pre-competitive academic industry consortium established in 2016, of which Merck was a founding member. It includes a collection of Finnish biobanks, the Finnish health system, and 13 industry partners to understand the genetic diversity of 10% of the Finnish population, which is 500,000 individuals, who also have national health register data that includes sort of hospital data, drug data, and primary care data. Right now, they have measured almost 1 million different SNPs designed to capture genetic variability in the Finnish population, which we now leverage genetic association analyses as well as meta-analyses with different biobanks. It's a 10-year project of which we're in the last 4 years here, and we currently have a sample number almost reaching the 500,000. And as you can see on the timeline on the right, the number of associations has gone up over that time with the number of gene disease connections being made. And really, we leverage this because it's a isolated population where some of these particular variants that are enriched in Finland due to the bottlenecking, and you can see here on this PCA plot that it's a unique population from the rest of the population surveyed here. And that on the right, a number of these variants discovered due to this effort are novel and are Fin-enriched variants that have been detected by their association with disease. So just wanted to close by one of the other advantages of this FinGen biobank, and I think one to look for in other biobanks is the ability to rapidly interrogate this data using a browser. And so this just represents a quick lookup of the FTO locus that I mentioned earlier, where we can look up specifically in this Finnish population what those associations are. And here you can see again the consistency of being associated with obesity-related traits, but really being able to quickly survey these results once they're produced is one of really the key elements of a biobank, I think, from our perspective. So just wanted to thank you very much, probably over time here a little bit, but really that we're pursuing drug targets supported by human genetics evidence because they do have a significantly higher chance of success, both by efficacy as well as safety. We really are leveraging these advances, some of which I described in genetic variant detection, to enable deeper insight into human biology and disease. And we really, though, still need orthogonal methods in order to validate some of these findings, in order to link GWAS to the causal gene with the impact on the protein function, and of course, to the link to disease biology. So look forward to discussion on any of those topics. Thank you.
Thanks. Thank you, Simone. So I think just for the audience, the chat function should be enabled, so you could put some questions in there, and if there are any specific ones, we will try to get to them. As I mentioned, there were questions that were pre-submitted by participants when they registered. So, I mean, I guess the question is, you mentioned, I think there was a mention of the UK Biobank. There's a mention of the, the FinGen, which are both, these 500K kind of samples and general populations. Does Merck and pharma industry in general, like how would you work with, or are you interested in working with smaller biobanks that might be more indication specific? How are you integrating those types of biobanks into your work?
Yeah, definitely. I mean, so I guess the first point is that, being able to replicate findings in one biobank in another is a key to validating the finding off the top. But then, of course, nowadays, 2 points. One is the disease biology link requires a deeper, I would say, clinical understanding of disease, which some of the smaller biobanks with a disease-specific focus, I think, is now, really of interest. That we have these sort of high-level findings from a population scale with sort of shallower disease or clinical information with something more deep where we can stratify subsets of disease. And then more importantly, the second point being progression of disease. As I think Kari nicely highlighted, right now we have some understanding of genetics with respect to disease risk. But at the point at which we actually treat a patient with a therapeutic, we're really treating the progression of disease, and whether what we understand from genetic risk of disease correlates with progression, as Kari already showed a story of that, not necessarily being true in one case, whether it is across diseases the same or not is still an open question, I think, and that we would really like to understand that better.
Right. So I guess that's, we talk about the advent of this field was all in genomics, but it's the integration of transcriptomics, proteomics, and I think Alexandre later will talk a little bit more about metabolomics. Is that, from a pharma perspective, is that kind of where you guys are going also in terms of the integration of all of the omics? And is that from a biobank capacity, I guess, is that one of the things, is the different types of samples and the ability to generate all this types of data for the different omics?
Not only omics, but also the sort of, I would say, the deep clinical information in order to understand the actual characteristics of a patient with disease, on top of things that we can test, right, in a biological sample, as you said, with omics. Yeah. Right.
And I would imagine potentially abilities to recontact and reintegrate or reinterrogate for different questions, it would be important.
Right, especially for progression of disease. Where that.
Right. Okay. Well, Simone, thank you very much for taking the time to talk with us today, and really appreciate it and your presentation.
Yeah, thank you so much again for the invitation.
Thank you. So, following along the theme of the theme of Canadians that are now kind of working with global groups, I'd like to introduce Danielle Larocque. So Danielle is with the Sanofi vaccine business, but is originally from Quebec and has, did some his original schooling here at, I think, McGill and then the Université de Sherbrooke before moving on to join Sanofi. So today, he's going to be presenting kind of main challenges to put in place for translational, science-driven trials and talk about biomarkers for vaccine development. So Danielle.
Hi, everybody. I hope you could hear me well. Yeah. So I'm today I'm located in Boston area because we have also at Sanofi a vaccines hub in Cambridge, in Cambridge area in Boston, and also especially in Waltham. So I'm presenting on behalf of Sanofi Vaccines, and also Steve Poirier, my colleague, is with me, especially for Sanofi Pharma side, that it will be able also to address a few questions on your point. I don't know if I have the control of the slide or you turn the slide for me, Jesse, how you want to work there?
If you just say next, I think we can close the slide.
Okay, good. Because related to what I've seen, I've changed few slides last minute. It's up to you guys, to accept it or not. Or there we go. I could
All right.
I will
We'll
try
see about
to,
sharing.
if it's, l'animateur a désactivé le partage d'écran par les participants. Okay, I could see that. I could not share my screen finally.
Okay, well, let's see if they can,
Yeah, we could not share our screen ourselves. It's just
they-
because related what I've seen, I could see there's certain scientific aspect that you would like to see, especially on
I think
biomarker assessment. So I adapt my slide for you guys because I know we have 125 participants, and I believe there's a lot of people coming from academic and coming from CRO as well that needs to know what are the needs when we work with biopharma in terms of biobanks. And let's say, especially if you're an academic players, and I could see, from Institute Cardiology, they have like a healthy donor biobank, and they might know, okay, what they need, like to as requirement to work with biopharma like us. I could see there we go. I could share my screen now. Thanks a lot to give me, and let me put that on full screen for you guys. Yeah, okay.
Perfect.
That's good. Okay, just to let you know, of course, Sanofi, it's a big industry all over the world, but in terms of research and development, like the main hub for Sanofi, let's say pharma, there's of course in the US and in France. And for Sanofi vaccines, especially the big hub is in Lyon, where I'm located most of the time. Marcy-l'Étoile, that's the R&D site. And also we have an R&D site in Canada, which is a little bit more on the development side than the research now, since 5 years in Toronto, where we have like over 2,000 employees there. And also, especially the research and development early, such as the mRNA vaccines in Waltham, Massachusetts, around Boston area. So, of course, as a global health company, we have like this business unit related to different type of disease. We have the specialty care, which Steve could talk about a little bit more than me, where immunology and biomarker assessments are really key for certain disease. We have the general medicine. As you know, we're a world leader on diabetes and as vaccines as well. Especially at this time, like you saw, like what we've been able to get, on subunit vaccines with GSK, with adjuvanted vaccines on COVID, but of course on flu, we're a world leader, and also especially the, in terms of the infant, the vaccines again, for the infants. Are really key for us, and also the consumer healthcare business unit, as you could see. So all of this business unit are needing biomarker assessment, especially at early phase, but not just in the early phase, also as post licensing and even in immunosurveillance as well. So there's various platforms that we're using at Sanofi, which are now a little bit more bio platform, as you could see, especially the antibody platform related on, like on pharma side, where we do with monoclonal antibodies, bispecific, and also domain antibodies, and also the nanobodies that has been invented, by Ablinks. That's part of our company. The nanobodies are coming originally with the hyperverbal region, from the camel. So we have this technology that we could target and develop antibodies rapidly. And of course, in terms of there's other technology that are coming a little bit more, as you could see, the PROTAC, such as the protein degrader, and also like even the artificial amino acids, such as the Centaurin platform, and also we have an NK platform as well as mRNA platform, as you could see, such as what we have in Waltham. So all of this platform are needing biomarker investment. And of course, in terms of vaccines business unit and vaccines platform, as you could see, we're located all over the world, especially in France and in the US and in Canada. And of course, we invest a lot in research and development where the biobanks are really key, like to address the question, especially on vaccines. So I'm going to talk a little bit more about vaccines today because, it's my playground at this time. And as you could see, we have a foundation of vaccines registration, especially like in the pandemic registration since COVID, and also like for flu vaccines. So we have a need to collaborate with leaders in immunology, such as what we do at this time with certain researchers over the world, as listed over here. And also we are interested to discuss also with other researchers in Canada. And we need also to have this new approach, especially using the machine learning and artificial intelligence, using biomarkers assessment coming from biobank to do this multi-parametric analysis with different type of statistic that never been used before, to see like the meaningful and the signature of certain biomarker, such as for vaccines, the vaccine responsiveness biomarkers, and even the correlative protection biomarkers. So that's really key. And of course, with the mRNA vaccines platform, that's the same thing as well. We need to optimize the sequence using certain system and software and artificial intelligence analysis to really be able to get the best sequence, not just for the expression and codon optimization, but also in terms of immunogenicity and even reactogenicity, because as you know, there's certain nucleotide of mRNA sequence that are more prone for reactogenicity than the others, as we have seen, for the COVID vaccines. So it's really key for us to have partnership with platform that could get access and to create also, and also are creating, biobanks related to this type of scientific question. So for us, if we work with biobank, as you could see for vaccines, infectious disease, we need this type of sampling, such as sera, PBMC, RNA, plasma, DNA, even a genetic variation now are evaluated by vaccines company as well, not just by pharma oncology company. Clinical observation, like this famous diary card information that could be used, for biomarker assessment, are really key, especially during clinical trials and even a post-licensing as well, like such as pharmacovigilance database, clinical protocol. So we need the information about the clinical protocol that has been used to create this biobank. The consent form, the ICF as well, are really key for us, to make sure we're compliant with different policies, such as the RGPD policy and the anonymization of the data. And of course, other type of sampling as well that are coming a little bit more important, such as for mucosal immunology, such as saliva. So as you could see, for us, the main objective is to find correlative vaccine responsiveness or immunogenicity, correlative protection, biomarker analysis. So for us, the main return of investment of this is to build on existing sample for assay development. So that's first things to get ready for new clinical trials. So we need in advance to work on assay development, and having access to a biobank, it's really key. Preventing creation of new clinical protocol as well. Instead of, especially when we're in assay development, instead of creating a new clinical protocol just for this, if there's a biobank existing over the world that we could get, let's say PBMC for cell-mediated immunity assessment, it will be a good return of investment for us to invest in this type of biobank, as you could see. So of course, we use this biomarker assessment and biobank for mechanistic question and answer. And most of the time, especially in our pre-IND, especially that section 2.6 on mechanistic question, it's really key to have this biomarker assessment. So having access to biobank, it'll help on this. Finding new signature or biomarker to be explored or validated in future trials, that's going to be really key for us as a return of investment, of course. And of course, for us, it's to facilitate future trials. Let's say we're able to find or correlate of protection or correlate of vaccines response. And instead of doing, let's say, multiple assessment, if we could narrow down just few testing in our clinical testing, because upfront, before we've been able to evaluate with biobank, let's say a biomarker of immunogenicity, let's say at day 7, that could correlate with protection at day 60 or immunogenicity at day 30. That's going to be a bona fide impact for us. So that's really like the main objective we want when we want to use biobanks. So for us as a requirement, there's 9 requirements for us. First, as a biobank, we need an institution review board approve protocol where that biobank has been used, let's say from our hospitals. Second, as I said before, the consent form needs to be clear for exploratory endpoint, because often we want to do biomarker assessment and they're considered exploratory endpoint, and they might not been put in the protocol and the ICF, as well as the agreement that the patient sign, before giving their blood, and so it needs to be clear to make sure we could do that. And of course, it could be, we need the information on the clinical protocol if phase 1 to 4 as well. So even like at post licensing as phase 4, we need to get access to the clinical protocols. It needs to be in agreement with human biological samples policy, like every company such as Sanofi, GSK, Merck, and others have internal policy in terms of human biological samples. And so it's really important, like to be in agreement with that. So we have to, even like for external sponsor study coming from academic, we need to be clear on this. And of course, the General Data Protection Regulation, the GDPR, even on not just on the samples, but on the data themselves, it's really need to be clear. And even like in different countries, we could get samples coming from let's say US going to France or vice versa. We have to make sure the RGDPD policy are respected in every country. Of course, an MTA agreement, a material transfer agreement, needs to be agreed, upon the parties. And of course, a data and samples retention plan. How long we could use these samples? Let's say when we receive samples from biobank, it needs to be stated, and we also need to prove after that when we destroy the samples, like we need to get that proof as well of the data retention. And 8, of course, the intellectual property plan endorsed by the provider. Let's say there's certain discovery that are done from this biobank, so it needs to be clear that what that data going to belongs to, who's that data going to belongs to. So that's really important to have that clear. Most of the time, like academic biobank are used for research purpose after that. So there's no need to have like intellectual property, let's say from the buyer of the biobank, but still we need to make sure of this like before buying these samples from this biobank. And 9, it's the data analysis plan. So the DAP, what we're going to do with this data. So we have to put a plan before how we're going to analyze the data from this biobank, how we're going to analyze, the samples, which type of readout going to be used. So most of the time we have to do that type of plan as well that are agreed among partners, especially if we want to do like more ad hoc or post-hoc analysis, from statistical point of view. And one of the good example we use over the world, and I hope Canada will also embark there, it's that famous UK biobank system where they really create like an easy to access system where you could apply. There's an application form you could get from the web or from in UK, where you have to state what's going to be your research plan using these samples from that biobank, even like at LT cohort. And of course, after that, you need to have this MTA and all of this amendment that needs to be required, to get access to this biobank. So that's a good example that we could look at in Canada as well. And the good things of on their system, they create also powerful tools to manage the data as well, which is called the UKBabe REST. So as you could see over here, they are able to do a little bit more multi-parametric analysis coming from all of this data coming from Biobank, from the UK Biobank. And we could also leverage that type of system on data analysis, to do comparison with other data from other partners that they could put in this biobank information. So that's going to be really the key in the next year, to get to also do this type of what UK did also with that biobank, and we hope in Canada that's going to be important. This is just an example of what we do for us on vaccines research. Of course, for us, the immunology is really important. The innate and adaptive immune system assessment are really key. So we need to have sampling that are able to get access to analyze in this time point post vaccination and even before vaccination. So the baseline status is really important. So we do this many readouts in terms of immunology for vaccines, as you could see, the cytokine, the cell-mediated immunity, the T cells assessment, the memory response. So all of this are really important. And having access to biobank that could help to do the assay development on these tests before clinical trials, it's a plus. Or having a biobank, let's say from a vaccination campaign, and having sample, let's say at day 2 or day 10 or day 7, it could be really important. As you could see, this is an example in our case where we like to have sample, let's say day 1 or day 2 post-vaccination or before the vaccination as well. And we evaluate certain type of cytokine with different markers, as you could see. So having also data coming from multiplex approach, such as Olink or Simoa technology or others, it's really key. There's more and more multiplexing technology in the field. So why not having the data also coming from the biobank on this, or able to do this type of analysis ourselves at biopharma or by academic partner. So that's basically like one examples of this type of things, even like on system serology and PCR analysis. That's really key for that. I don't want to go too much in details, but like another example that we have done, and it's a study coming from 10 years where they have done a biobank at Institut Pasteur Paris. It's called the Milieu Intérieur Project. And as you could see, they've been able to have a 1,000 subject that they provide multiple samples there. It's like nasal swab, blood, stools, and even skin biopsy for certain, where they provide, like a sample throughout at least 2 to 3 visits. And they follow this subject, for 10 years for different type of analysis. And of course, during 10 years, the technology are changing. So you need to get access to this V0 and V1 visit, even, after 10 years, let's say if the technology is evaluating. And so that's a cohort to understand a little bit more, let's say, the healthy status of a population in France. So they're doing a lot of biomarker assessment with different markers by flow cytometry. So that's called that famous Milieu Intérieur project that started from 2012. And now they are in the step, to do the last visit 10 years after, coming from a beginning it was 1,000 subject. Now they have over 600 subject. So that's an example of a biobanking study, I would say. So thanks a lot. I'm welcome to answer any.
Yeah, I mean, I'll ask you a quick question, but we're a little tight on time, but excuse me, it was interesting that you were talking a little bit versus, the earlier talks about, sample as opposed to just data from the biobanks. Is that, particularly challenging for biopharma to be able to access or to be able to actually get samples as opposed to data from these biobanks?
Yeah, especially like the biopharma are asking, like samples coming from this biobank, not just for assay development purpose, but also for mechanistic purpose as well. So yeah, there's a need, and especially when we have research center coming, where let's say like Sanofi have a research center in the UK, and of course for them it's easy to get access to the UK Biobank. So same thing in Canada for us. Let's say if we have an asset development to be done in Canada, and we could get access to samples that could come from Canada without having to ship, over countries. That's a big need. Yeah, exactly. On top of the data.
Okay, thank you. All right, thank you. We're a little tight, so I'm gonna say thank you very much for presenting, and I'll introduce the final speaker of the webinar, which is Alexandre Leboutillier. So Alexandre was the co-founder of Imagia, which was leveraging AI and to accelerate the accessibility and development of precision medicine, and now is working with a group called Linearis, has helped found Linearis, and will give us a quick talk on what Linearis is doing and how it's moving forward. Alexandre?
Thank you, Jesse. What I can say is that we live in an interesting time with all those omics and clinical data. I'm sure there will be interesting discoveries and acceleration in commercialization of those discoveries in upcoming years. So today I'm going to cover two topics, mainly how we can apply AI in biobanks, and specifically what kind of data and metadata are useful. Essentially, the Linearis lab is an industrial lab that we're creating based on the experience that was gathered from 9 academic labs in Canada. So it's going to be an industrial labs with certification focusing on metabolomic disease. So cancer, diabetes, infectious disease, NASH, cardiometabolomic disease, and essentially those discovery could be useful for earlier diagnostics and drug discovery in AI. The investment thesis and what we're focusing on is to increase the understanding of disease model. That's the whole goal of causal model in AI. So there's a lot of literature, and as well as a software pipeline that we have developed that, in our opinion, increase the understanding of disease model. And interesting fact to know, the FDA since January no longer requires animal tests. So this is a real opening for organoid testing and working more with artificial intelligence and in silico pipeline. The lab on the left side, as I mentioned, is focusing on metabolomic analysis. So the UK Biobank, everybody talks about the UK Biobank, started those analytics. About 112,000 samples were analyzed, not on a full panel of metabolomics, but they were able to find 4 disease signatures. So the trend is starting. People want to look more at metabolomic data. So the lab is focusing on this type of data. But of course, you don't want to have a narrow vision. We take into account other type of data, including genomic and proteomic. And those data are focusing in discovering disease signature. I'm going to explain to you in the few next slides what do we mean by disease signature, but you can understand that once you have a better understanding of what is the disease, it's easier to support those 3 types of company through the fund that we're launching, Linearis Venture. I would say for the past 8 years, I've been working on genomics, on transcriptomic, on imaging data, and the one data that from an AI perspective generate a lot of data that is as close as possible to the phenotype is really the metabolomic data. Those are the small molecule of life that do manage what you eat and transform them into energy or matter or expel the waste. So when you have a metabolomic disease such as cancer or diabetic, of course, the metabolomic data or analysis will be unbalanced. And there's documented pathways David Wishart, which is one of the respected individual in that field, created a database called the Human Metabolomic Database. There's currently 200,000 documented metabolites for which we can have the associated pathways. There's also small collection device. Many of you are familiar with many companies such as this one, Tasso. They produce IVD certified collection device. We've used these during the COVID pandemic to collect immune response to the vaccination at 2-hour interval. And that's very interesting because you can have a very granular effect on the metabolomic data at a very cheap cost. It's around $5 per dose device. This is the type of workflow, very simplified. We use those data that are collected at home. So patient can send them the next day on mass spectroscopy. And I would say we were stopping there at the third pictogram where you see the metabolites popping up about 5 years ago. But with AI, we can make a sense of all those data and come up with a metabolomic signature for metabolomic disease. And I'm going to give you the example of COVID. During the COVID, we've access to a small cohort of about 5,000 patients. Each of those patients have about 33 clinical variables. So this is a good number when you are in clinics, including imaging, systolic pressure, but none of those variables were clinically sufficient to make a diagnostic. But there were about 6,000 proteomics and metabolomic markers. You see them on the axis, on the x-axis, and some of them in blue. And I'm going to zoom out on those metabolomic marker. They were significant enough to characterize even a proteomic disease. So that's very interesting to see for this kind of disease that there are unbalanced metabolomics. You see those on this table, you see the documented pathway or sub-pathway, and you see if they're up or down. The blue one really characterize the disease, and that might be very interesting to see if you have the long COVID, will those markers still hold? So once you have those metabolomic disease signature, that could be in lung cancer, that could be in other type of disease, there's documented pathway that we can incorporate into what traditional AI drug discovery are doing. They have an optimization function and they have a generative model that do generate a compound of interest. But typically this optimization function and this generative AI are focusing on either derivative of AlphaFold for protein binding or trying to block specific pathway, but they're not using, I would say, a more humanized model. So this is why it's very important to add more data to this optimization function. And what's really interesting with metabolomics signature is once you test those compounds in a cloud lab, I'll come to that definition later, you are able to measure at a granular level what is the impact of each of those metabolites in a granular function. So you can send that back to the AI model to refine it in a way that you can do that iteratively to increase the understanding of the disease model. Furthermore, once you have that disease signature, and as I mentioned, the FDA no longer requires animal testing, I will see an increase in the market of organ on a chip. So there's many technology where you can have a specific organ, epithelial or hepatic cell line, and you can modify those with CRISPR-Cas9 technique and use either existing drug or the new compounds that you have developed and go back on the mass spec and see the impact on this organ with the metabolomic response. For example, we are working on a NASH disease. We made one mutation on this organ, and there was more than 150 metabolites that have changed. Of course, when you're testing new compound, you can see the impact of this mutation. So why am I going into the detail of this disease signature is I think there's an appetite for biobanks to improve the data that they have, so to digitize more omics data, so metabolomic being one. But it's important that the data that you have in your biobank contain metadata and in a format that can be easily ingestible by AI model. So we have a pipeline that do transform those type of data, typically saving 3-4 months of just cleaning the data to automatically import it into the AI model. Previously, in the discussion, Simone mentioned that certification is very important. Not on this slide, but we're developing a certification for 1,400 metabolites in collaboration with other metabolomic labs in the world. So if you run those samples, either in Japan or in US or in Europe, in those labs, you should have the same result or compatible result. Those are just examples of organ on a chip. Again, testing that disease signature that you would find on your biobanks, and you find various type of imaging that are very useful to understand what's happening with the compound that you're injecting in those organ on a chip. So the idea is to move from the biobank to digital data of metabolomic data to a digital signature and use those digital signature of the disease in the pipeline of research. The most important aspect of analysis is consistency, certification, but also high throughput. If you want to do AI, you want to do a lot of experiments on your sample and a lot of analysis. So we're working with a company that increased the capacity of those mass spec to about 250,000 sample a year per mass spec. So the capacity is there, and you can do the sample of the biobank, but also the sample of organoids, which is a two-pronged approach to see the disease signature, but what happened when you're testing the disease. Once you have that signature, you can apply this signature both at the drug discovery, at the prevention, and also the early diagnostic size. I'm going to move quickly to the core team. David Wishart on metabolomic is the key person, manager of the TIMIC in Canada and creator of the drug bank. Joshua Bengio, which always helped me in all the AI project. I have a lot of interest in drug discovery, so that's why we want to push the drug discovery format on type of data so they're useful for the AI. Manon Boisclair, lots of experience at the global level, and Jacques Corbeil, holder of the Genomic Canadian chair. Thank you very much for your attendance to this webinar, and I hope we can continue the collaboration and discussion on how we can combine genomic, proteomic, and metabolomic to accelerate the discoveries.
Thank you very much, Alexandre. Let me just pull something that if there are any specific questions. So I think you know it's interesting to hear you talk about it's metabolomics in general, but the implications of AI. And I think with the biobanks I think that's something we'll see more and more is the integration of the utilization of AI for this data. But as you said, I guess the challenge being older biobanks that were set up without an AI kind of point of view. Is that a major challenge for data banks or biobanks, sorry, in terms of having AI accessible data?
Yes and no, because AI can really help to clean that data, but the pipeline is important. So that's why we focus a lot of our effort in developing that pipeline that can convert, I would say, traditional data that come out of a mass spec to data that you can just import in your model in the matrix format. So I think there's an evolution. The metadata sometimes are not captured, and when it's not captured, that might be too late. So that's why it's important when you do the analysis, do it once, but do it in a proper way. Pharma like to have certification when those analyses are done. So of course, we don't want to spoil a sample and have to redo it again.
Okay, wonderful. Well, we're a little bit over time, so I thank you to everybody who's kind of stayed with us. I would say thank you to our presenters, Kari, Simone, Danielle, and Alexandre. Again, thank you all for all of you who have participated. And we'll be sure we will be back I'm sure in the fall with another webinar, and I look forward to hopefully having you join us then as well. Take care, everybody.